Alright, so splicing in this video. The splicing removes the flanking of the intron that's between two different exons. And the spliceosome is the machinery that carries out the splicing, and there's two transesterification reactions in this, which we'll go over soon. There needs to be a very small amount of conserved sequences in the intron for correct splicing to occur. So if the intron is supposed to be 10,000 bases long, but this is only 2,000 bases long, it's okay sometimes, most of the time, and it won't affect the genes that are in the exons. The introns, they can be 50 pairs, not base pairs, but bases long to 10,000 bases long, or nucleotides long. And so, like I said, insertions or deletions in the introns do not affect splicing in most cases. Alright, so over here we can see that all introns begin with GU and end with AG. So the introns begin with GU and end with AG. There's the pyrimidines. There's 10 pyrimidines, and then any nucleotide, and CAG. That's the normal 3' end of the intron, and the 5' end. That's the sequence of the 5' end of the intron. So the 5' end is called the splice donor site, and this is called the splice acceptor site. The 2' OH on the adenine, we'll see later on, that's what's going to attack the intron on the 5' side, and then the free 3' OH group on the exon on the 5' end is going to attack the 5' phosphate on the downstream extron, or the one is on the 5' side. So we're here showing the reaction. So this adenine over here, it gets displaced because of U2. U2 does like base pairing with the sequence at the 3' splice site, or the splice acceptor site, and the base pairing is bit slanted over here, so this adenine is kind of free. So that's why this adenine can attack this U over here. The adenine's 2' OH is gonna do a nucleophilic attack over here, and then the intron over here is gonna get cut, and then this G goes over here, and so it's gonna make like a lariat like this over here, and then this U is gonna have a free OH at the 3' over here, and then it's gonna attack this this 5 prime end of this U over here. So this is going to do a nucleophilic attack over here, and then you're going to get the spliced RNA. And then so, well, it's going to have three phosphodiester bonds, this adenine, which creates a lariat. And then there's a lot of SNRPs involved in this reaction. There's SNRPs are small nuclear RNA, which are associated with proteins, and they form SNRPs. And they complex with other proteins to form the spliceosome, which is the machinery. Like over here, we saw spliceosome is the molecular machinery. We carry out the splicing and consists of two trans-exterifications. The two trans-exterifications was this nucleophilic attack by the adenine over here, and the nucleophilic attack from here to there. Those are the two trans-esterifications it's talking about over here. Okay, so the spliceosome assembly starts with U1 binding to the 5 prime splice site here, and U2 at the branch site, and there's ATP used over here for the U2 to bind over here. And then over here you see this complex U4, U6, and U5 complex coming together over here, and then this U6 is going to dissociate with this, and then this is going to leave, and then the U6 is going to come and displace U1, so you lose U4 and U1. Over here U4 and U1 are leaving. There's some more ATP being used and converted to ADP, and it brings this close together. And then this adenine, the one that's gonna have the 2 prime OH group, is gonna come and attack this G over here, and then it's gonna leave that free OH from this side of the exon, and then that's gonna come and attack this piece of exon over here. So basically you have U1, U2, and 4, 5, and 6. So basically everything from 1 to 6 except 3. Don't have to worry about what 3 does. So 1 is at the donor site, I think. Yep. Donor site, the 5 prime donor site. And 5 prime donor site is U1, and in the branch site we have U2, and we get in between the donor site and the acceptor site over here we have the U5 and U4 and U4 U6 complex. And then so basically U6 and U2 are involved in the trans-esterification reaction, which is gonna occur here. And this is just showing the dislocation of the adenine because of the U2 binding to the intron in this manner over here, and this adenine is going to come and attack this adenine over here, and then this exon is going to attack this exon over here, and you know, yeah, so then you get this complete exon. They act as represent the EU6 and U2 catalyze both transesterifications. We talked about this. The U6 detaches from U4, U6 from U4, and then goes and displaces U1. Oh, and then this consensus, they do the binding, the U1 over here, and then you have U2 doing the consensus. They're doing the hydrogen bonding over here. That's how they find the introns. So then if you had a mutation in the AG or the GU, then you would mess it up, and then that would lead to abnormalities. But if the mutation was somewhere over here, it wouldn't really affect it. Over here it might not affect it also, but exactly at AG and GU. So like if it's over here AG, then this would bind maybe over here instead of over here, and then you have these extra nucleotides and you'd have an extra amino acid or something, so that would cause a mutation. But if this one U was different, might not have a big effect, or maybe if this U had, if this A was different, might not have a big effect. But if you have AG or GU changed, there would be an insertion or deletion. So mutations here would result in abnormalities.